and shadowy heart of Delphi's forgotten necropolis, archaeologists unearthed a cracked obsidian tablet inscribed with a language older than Olympus itself. Whispers say it carries the genetic blueprint of demigods, hidden instructions encoded in our very DNA. Tonight on the podcast, we'll retrace the steps from the silent tomb to the molecular laboratories where scientists race to decode the tablet's myth secrets. Are you ready to unlock the ancient code that could rewrite humanity's origin story? Greetings, friends! Greetings, cosmic voyagers! I am Kealion Bay, welcoming you to the micro-DNA lab nested in the bowels of our stellar biome starship. As a kid, I gazed at the stars and asked, are we alone in the universe? Today, I've discovered each of our cells is its own mini-star factory. In this podcast, we'll unveil humanity's hidden universe, delving into DNA's coded cosmos. Ready? Strap in and fire up our molecular rocket! We won't settle for dusty textbook definitions. We are rocketing to molecular biology's frontiers, feeling each fact as if charting new star maps. Stay tuned until the end of our special Brainwaves coupon. Grab my comprehensive Udemy course, SAT Biology 101 Unlocking Life's Blueprint Level 1 at a discount, and receive a 3D printable molecule model at launch your endoscope exploration. Our molecular journey's first waypoint, the DNA double helix. Those spiral that mesmerize you. They are life's master blueprint, but what holds those spiral together? Holographic lab sign. Activate. First of all, nucleotides and base pairing. Each nucleotide comprises a phosphate, a sugar, and a base. Adenine A, thymine T, cytosine C, or guanine G. Adenine pairs with thymine, guanine pairs with cytosine. Think of them as alien civilizations sending sacred handshake signals. Perfect hits across cosmic distances. Why AT and GT? I mean, adenine to thymine and guanine to cytosine. How do weaker or stronger base pairs affect helix stability? Let's talking about helix stability and hydrogen bonds. Adenine and thymine pairs share 2 hydrogen bonds. Guanine to cytosine pairs share 3 bonds. Regions rich in guanine to cytosine boast extra stability, like reinforced starship hull segments braving cosmic storms. Close your eyes and envision the trillions of helixes in your body, each safeguarding your life's code. How do they endure the molecular void? Universal genetic language. Let's talking about universal genetic language. All life on Earth, from bacteria to humans, uses nearly the same genetic alphabet. A bacterium on one world and a plant on another speaks same letters. This hits at life's deep interconnected web. When we hear self-replicating machine, we imagine sci-fi wonders. Yet DNA perfected this eons ago. Let's replicate it. Listen to the cell division symphony. First of all, unzipping the helix. Helicase enzymes unzip the DNA like a molecular zipper, akin to opening a ship's airlock. Helicase unwinds 50 to 100 base pairs per second. Imagine reading 100 letters per second mid-flight. Oh my God. I need to drink coffee, voyagers. RNA primers launch. DNA polymerase needs a primer. Short RNA fragments ignite synthesis like rocket ignition sparks. Polymerase's precision. DNA polymerase reads the template 5 to 3, adding matching nucleotides at approximately 60 per second. A human cell must copy 3 billion base pairs before division. That's billions of letters shorted every hour. Oh my God. Okazaki fragments and the lagging strands. The lagging strands synthesized in Okazaki fragments joined by DNA ligase. Each fragment is a precise puzzle piece snapped into place. Next, the genetic code, triplet codons translate into amino acids. 64 codons encoding 20 amino acids orchestrate protein diversity. First of all, the codon table. AUG starts translation with methionine. UUU specifies phenylalanine. Picture cosmic data rockets roaring amino acids shipments. The second is start and stop signal. So important, so significant. UAA, UAG, UGA, halt translation like an emergency ducting command. Mistakes here can cascade into molecular catastrophes. Translation mechanism. Ribosomes read mRNA, tRNAs deliver amino acids like robotic arms, and on assembly line forging living machines. And a single point mutation, one letter change, can rewrite entire molecular novels, sometimes unleashing disease or profound adaptation. Intrigued? Imagine an equatorial space station colonized by aliens needing custom DNA, designing plants and microbes thriving in microgravity. CRISPR emerged. First of all, biotechnology frontiers, gene editing, biofuel producing microbes, next-gen vaccines, molecular wizardry reshapes industry. Ethical horizons. Could we become a genetic elite? Transition from Homo sapiens to Homo technologicus? These questions leave deeper marks than any star chart. Our journey into DNA's hidden universe ends here. Especially this topic ends here. I hope you have felt the power of unrivaling life's code. But our adventure countries use code brainwaves. On Udemy's SAT Biology 101 Unlocking Life's Blueprint Level 1, grab your discount and 3D model gift. Dive deeper and join me next time for Protein Factory's Ribosome Wonders. Before talking, let's drink a coffee, Voyagers. Welcome, explorers, to the virtual conference chamber of our stellar biome starship. I am Kealion Bay. Today, we pry open the magic doors of biology's most enchanting factory, protein synthesis. Protein synthesis inside the cell. Are you ready? Then drop into your virtual belts, lower your voices to a cosmic register, and fire up our molecular engines. This podcast will feel like a sci-fi epic, yet deliver molecular biology tuition worthy of top-tier students. Don't forget code brainwaves for my Udemy course, SAT Biology 101 Unlocking Life's Blueprint Level 1, packed with 3D printable models and high-res graphics to bring the mystery to your desktop. Subscribe and show your support for space mission biology. Now let's dive into the cell factor layers. Transcription, translation, and the engineering marvel of ribosomes. Like any factory, the cell starts with blueprints. The nucleus is the common center. Here, the glorious DNA blueprint is transcribed into RNA. We call this transcription. First of all, nuclear envelope and entry positions. DNA is safely locked inside the nucleus. The nuclear envelope acts like our ship's airlock, allowing large molecules through under strict control. RNA polymerase waits for the go signal to bridge to lock. RNA polymerases dance. We call it the molecular scribe. It unwinds the DNA helix and synthesizes an mRNA strand. Imagine a cosmic typewriter firing off 40 to 50 nucleotides per second. As your biology replicates billions of bases, this scribe sits across the genome at blistering pace. Promoter-based initiation. Every gene start line is a promoter region, a start command for RNA polymerase. Like an astronaut, go ahead to open the airlock. How tight the polymerase binds here dictates gene expression levels. Another section is, I'm talking about elongation phase. A polymerase moves, it rewinds DNA and releases mRNA. Think of processing a film reel seamlessly until the stop call arrives. This dance flows uninterrupted. Termination and capping. At the terminator signal, polymerase stops. The mRNA gains a 5 cap for ribosome recognition and a 3 poly a tail for stability. Now it's ready to exit the nucleus and journey to the cytoplasm. Thus emerges a fully processed mRNA, an interstellar message primed for the ribosome station. C now shifts to the cell's colossal assembly line, the ribosome. mRNA has arrived. tRNAs line up. Amino acid blocks snap into a place to build proteins. Initiation complex. Ribosome splits into 40S and 60S subunits, assembling around mRNA and initiator tRNA carrying methionine. This is the initiation complex. Methionine is the universal start fuel, like priming a cosmic reactor. Codon-anticodon pairing. mRNA's 3-letter codons match tRNA anticodons. AUG, UUU, GGC. Each codon maps to an amino acid. Imagine 64 codons as galactic data packets directing traffic of biosignals. Peptide-bound formation, 23S. rRNA's peptidyl transferase activity links amino acids, releasing the previously amino acids. Think of a robotic arm fastening parts on a zero-gravity station. Cells from thousands of peptide bonds each second. Elongation cycle. A site Incoming amino acid tRNA docks. P site, peptide chain grows. E site, spans tRNA exits. This 3-site choreography is a subatomic ballad, precision engineered. Termination, stop codons UAA, UAG, UGA, summon release factors, disassembling the complex. The nascent polypeptide exits in its initial form. Folding and processing. Polypeptides fold into 3D shapes with chaperone proteins guiding them. Like industrial robots sculpting liquid metal, disulfide bonds and glycosylations deliver final functional touches. Thus, nanoscale factories forge and proteins fueling lyse machinery. Armed with these insights, what can we build? From CRISPR to biofuels, biotech is revolutionizing industry, machine, and space exploration. Recombinant protein production, insulin, vaccine antigens, industrial enzymes, all mass-produced in bacterial and yeast cell factories using these synthesis pathways. Synthetic cell factories, lab-engineer entirely novel proteins, space-resilient enzymes or microbes that convert waste into biofuel. These living machines transform raw materials into high-value products, future space colonies, portable bioreactors on Mars generating consumables, living modules at lunar bases supporting human crews. Soon our world habitats will thrive on engineered biology. Ethical and social dimensions. Editing human genomes or spawning new life forms demands responsibility. As scientists and planetary citizens, we must tackle these questions head-on. Our journey through cell factories, the perfect mechanism of protein synthesis, comes to end, but for now, I hope you feel the gears, the robot arms, the wonder driving molecular assembly. Coupon code Brainwaves, SAT Biology 101 Unlocking Life's Blueprint Level 1 on Udemy, my course. Download your 3D printable cell model and host your own nanofactory at your desk. This channel is the cornerstone of my scientific documentary dream. Subscribe, leave your thoughts, and join me next for Epigenetic Beyond Our Genes. Let's drink coffee, friends. Welcome, pioneers, to the galaxy's wildest lab. I am Kealion Bay, transmitting from the Stellar Biome Station. Tonight, we'll lift a veil on DNA's most controversial weapon, CRISPR. Try to sci-fi cliches about the gene hackers. Frustrated by sensational social media myths, I'll guide you through real labs, emerging tech, ethical sparks, and the world awaiting us, all in a thrilling sci-fi ambience. Remember, this channel is my biology documentary dreams launchpad. I'll bring molecular mysteries to your desktop with high-res 3D visuals. Use code Brainwaves on my Udemy course, SAT Biology 101 Unlocking Life's Blueprint Level 1, fueling our epic production. Subscribe and support, and together we'll unlock the universe's secrets. Buckle up, ignite our nano shuttle's rotors. We begin CRISPR's Sacred World Tour. CRISPR Cas9 sound alien, but it's nature's own genome editor. Let's decode the jargon. What is CRISPR? Clustered regularly interspaced short palindromic repeats, a bacterial immune library of viral DNA snippets, storing digital portraits of space pirates of instantly recognized repel them. Cas9 molecular scissors. Guided by gRNA, like GPS coordinates. Cas9 cleaves target DNA at precise sites. HNH and RUBC, the main snips both strands and nano-snapple remover slicing paper into. Mechanism overview. Synthesize gRNA, matching our target. Package Cas9 gRNA complex into cell via lipid nanoparticles or viral vectors. Complex locates the DNA target and cuts. Cell repair. Listen, NHEJ, blunt edits or HDR, precise insertions. Breakthrough applications. Gene correction from cataract-free rats to sickle cell therapy in human HSCS. Case study 2017 Chinese trial editing beta thalassemia in embryos sparkled heated ethics debates. Root and misinformation. Social myths. Super soldier. Designer babies. Reality. Off-target cuts. Low HDR efficiency. No lightsaber-style gene zaps yet. CRISPR is still crawling, but its potential to reshape worlds is undeniable. Even the brightest tech needs a moral compass. CRISPR forces us to ask, just because we can, should we? Germline versus somatic editing. Somatic, patient-only fixes. Germline, alters eggs, sperm embryos, heritable changes. off-target risks, undesired cuts can knock out tumor supersessors, causing chaos, access and equity. High-cost therapies risk widening the genetic divide, only the wealthy benefit. Feature scenarios: designer babies, demand for enhanced intelligence or strength, enter at dystopia. Biodiversity threats: CRISPR mosquitoes versus ecological balance, careful chain reaction planning required. These questions are our ethical questions. We can open every door with CRISPR. But can we relect them? Unleash your imagination. What will CRISPR achieve by 2050? Personalized cancer vaccines, mRNA vaccines tailored to tumor mutational profiles, training your cells to revolt against cancer. Transplant revolution, CRISPR-edited pigs yield organs compatible with human recipients, slashing rejection rates. Slowing aging, controlled telomerase expression might stall cellular senescence, balancing youth with cancer risk. Extremophile biologies, Mars-resilient plants, microalgae, thriving in zero gravity, terraforming essentials, biohybrid systems, living sensors, cyborg cells, merging electronics and biology, nanorobots, patrolling your bloodstream. These visions sound wild, but today labs sow their seeds. Our exploration of genetic engineering, how CRISPR rewrites lives, ends here. I hope you can now distinguish reality from hype. Coupon code Brainwaves. Use code Brainwaves on my Udemy course, SAT Biology 101, Unlocking Life's Blueprint Level 1. High-res 3D visuals and molecular models await. Subscribe, share your thoughts, and join our journey. Epigenetics beyond the genome, delving into layers beyond DNA. Don't miss it! Welcome, Voyagers, to the Epigenome control room aboard the Stellar Biome Station. I'm Kealion Bay, your cosmic bioarchitect. For millennia, humanity believed destiny was written in stars, overseen by supernatural force or zodiac signs. But what if the threat we call fate waves not only through the fixed bricks of our DNA, but through environmental furnaces, living causes across time? Tonight, we embark on epigenetics. Are our genes our fate? An odyssey where base pairs remain constant, yet gene expression soars on wings of chemical marks. Forget astrology. I hate it. I don't believe it, by the way. So forget astrology. Here, carbon methyl groups, histone cups, and environmental triggers. Write the script. This is no dry lecture. It's sci-fi changed, yet rooted in cutting-edge molecular biology. Stay tuned for code Brainwaves to unlock my Udemy course, SAT Biology 101, Unlocking Life's Blueprint Level 1. You'll receive high-production 3D molecular demos and 3D printable epigenetic models. Subscribe and comment. Your support propel our galactic research, collaborating with alien intelligences beyond. Fasten your belts, lower your vocal register, and fire our nano-shuttle's engines deep into the epigenomic layers. First, let's define epigenetics, the chemistry above the genome. The ATGC letters you're memorized in high school stay put, but which genes ignite or dim depends on chemical marks. Our cellular control panels. First of all, the gene expression dashboard. Every gene connects to a dashboard inside each cell. Epigenetic marks act like light switches, dictating where RNA polymerase can land. Example, the identical DNA sequence becomes a neurotransmitter factory in brain neurons, but a detox workshop in liver cells. The same code, divergent destinies. The core mechanism, methylation and histone modification. DNA methylation, when a methyl group CH3 tags a cytosine base. It shut gene expression sometimes for decades. Histone acetylation. Acetyl groups in histone tails loosen DNA's warp. Remove them. Chromatin tightens, transcription halts. Epigenetic inheritance. Nutrition. Stress toxins in the womb can program adult metabolic disease and behavioral quirks. In mice, specific histone marks pass across generations. I'm talking about a story. Babies born during the 1944 Dutch famine later suffered heart disease and diabetes. Maternal starvation penned chapter 1 of an epigenetic saga. Thus, to claim genes are destiny is to ignore epigenetic canvases and environmental forces shaping our fate. Now, plunge into nano detail. The chemical engineering being DNA methylation and histone tail modifications. DNA methyltransferase, DNMT. DNA methyltransferase, DNMT family. DNMT3A, 3B establish new methyl marks. DNMT1 propagates mark during cell division, like a digital backup system in a Starship core to prevent data loss. Target zones. CPG islands, clusters of CG pairs near promoters. A methyl flag here erects an immovable barrier to transcription machinery. Histone acetyltransferase, HATS, and deacetylases, HDACS. HATS add acetyl groups, neutralizing histone's positive charge, losing DNA histone grip, chromatin opens, genes on. HDACS remove acetyl, chromatin closes, genes off, impact memory formation. Neurological disease rely on this off switch. Additional marks about methylation, phosphorylation, ubiquitination. Histone residues can carry methyl, phosphate, or ubiquitin groups, expanding expression possibilities like tweaking chords in a cosmic symphony. A single note shift changes the melody entirely. Imagine chemical toggles that compose molecular novels, writing cellular fate one mark at a time. The real marvel, environmental stimuli, writing epigenetic scripts, not as supernova's radiation, but diet, exercise, toxins, stress. They echoes across billions of cells. Nutrition and metabolic signals. Methyl donors, folate, B12, methionine shape, DNA methylation. Example, vegetarian mice exhibit gene expression patterns resisting obesity. Thanks to altered methyl pools, stress and hormonal waves, cortisol surges, trigger histone modifications in hippocampal genes, altering memory and emotion pathways. Case, offspring of the World War II veterans showed heritable methylation change on stress-responsive genes. Toxic exposures, air pollution, heavy metals, endocrine disruptors, leave indelible marks on fatal epigenomes, shaping immunity, metabolism, and hormone balance. A research babies born after Fukushima showed aberrant methylation in metabolic genes, physical activity, and lifestyle. Exercise sparks histone modifications that upregulate anti-inflammatory genes. Health living is an epigenetic manifesto. Daily choices are brush strokes painting a dynamic portrait atop our genetic blueprint. Genetics isn't just academic, it's revolutionizing medicine, agriculture, or even space travel. Disease, diagnosis, and treatment. Biomarkers monitor prompter methylation in blood, urine, or salvia to detect cancer and neurodegenerative disease early. Therapeutics, HDAC and DNMT inhibitors in cancer aim to erase pathological marks and reset cells to normalcy. Agriculture and food security. Epigenetic engineering crafts drought-tolerant, nutrient-rich crops, crucial of Martian greenhouses. Aging and regeneration. Induced pluripotent stem cells, iPSCs, use epigenetic reprogramming to rewind time, generating youthful cell profiles. Spaceflight and human adaptation, microgravity and cosmic radiation rewire the epigenome. Future astronauts may use epigenetic drugs or training regimes to shield against these changes. Genetic armor for the cosmos. Epigenetics guides the hand that sketched atop our genetic script. Here, true miracles take shape. Our exploration through epigenetics, are our genes, our fate, concludes. I hope you now see DNA not as destiny's cage, but as the foundation for a dynamic, interactive masterpiece. Coupon code BRAINWAVES. Use this on Udemy for SAT Biology 101 Unlocking Life's Blueprint Level 1. Download the 3D printable epigenetic model and bring molecular science to your tabletop. Subscribe, leave your thoughts, and prepare for our next voyage. Synthetic life, artificial cells, and new biological worlds. We'll chart nano evolution's frontiers. Don't miss the journey. Greetings, explorers of the cosmic geneverse. I'm Kealion Bay, your cosmic guide aboard the interstellar lab ark. Tonight, we stand on the edge of the unimaginable, synthetic life and artificial cells. We are no longer spectators. Soon, we'll be architects of living machines. carbon-based organs swirling like nebula in the petri dishes, or perhaps silicon-based constructs humming like quantum circuits. Yes, you heard correctly. Laboratories around the globe are forging programmable, stable cells, hybrid creatures that blur lines between machines and organisms. In this voyage, imagine creating a living micro-robot that pairs tissues, or a programmable microbe that deconstructs plastic in the Pacific garbage pact. This is no mere lecture. It's a living sci-fi odyssey, grounded in real molecular biology breakthroughs. Don't forget to use code BRAINWAVES for my Udemy course SAT Biology 101 Unlocking Life's Blueprint Level 1, complete with high production 3D graphics and downloadable microcell printables. Subscribe now and feel the dream of our interstellar bio-bridge. Fasten your belts, deepen your voice, and prepare to plunge into the nanocosmos of synthetic biology. What is synthetic biology? It's rewriting nature's source code. But how do we hack life? Here are our molecular power tools: modular gene parts, biocircuits design, minimal cell frameworks, and in vivo testing platforms. Let's decode each pillar. First of all, modular biology parts. We thread DNA segments like Legos, promoters coding sequences, terminators using Gibson assembly or Golden Gate cloning. We snap together genes with robotic pipettes selecting parts, and plate readers validating output in seconds. I tell a story. Picture a robotic arm. Pipetting arm guided by AI, choosing your favorite promoter, slicing in a fluorescent reporter, sealing it into a vector in all under 2 minutes. It's like BIOS rewritten by micro drones. Synthetic gene circuits. We build logic gates in cells, and or not, we design switches for biofilm production or kill switches that self-destruct cell under defined conditions. Example, in 2016, APFL researchers built a bacterial logic circuit that triggers cell death only when both low oxygen and high toxin levels are detected. Our microbial Sentinel awaits your commands. Minimal and synthetic cell frameworks like stripping and OS to its kernel. DJSVI Syn 3.0 minimal cell holds just 473 genes. The essential genome. By iteratively testing thousands of genes from complex organisms, we identified the bare minimum to sustain life. Think of it as a cell smartphone in airplane mode. Only the core apps run. Nothing extra drains resources. Designing life in the lab. We synthesize entire genomes in vitro and transplant them into host cells. In 2010, a fully synthetic bacterial genome booted up in a mycoplasma cell, giving birth to a Volk-alive organism. Case study at JSVI. Scientists built the Mycoplasma mycotes genome from scratch, inserted it into a recipient cell, and the cell divided, providing life can start from code alone. There you have it. DNA Legos, gene logic circuits, minimal cells, and the novel life design, the cornerstones of synthetic biology. Now, how do we assemble a true artificial cell? Let's engineer step by step. First of all, building a cellular chassis. We create liposome or proteoliposome shells, synthetic lipid bilayers mimicking natural membranes, encapsulating our minimal genome and molecular machinery. Picture a nano-starship hull infused with ribosomes, polymerases, and energy modules. Analogy: this is like fitting a starship bridge with navigation consoles, reactors, and crew cabins, all inside a self-sealing sphere. Constructing metabolic pathways. Even minimal cells need energy and biomolecule synthesis. We encode synthetic metabolic networks for ATP production, protein translation, DNA replication, using mathematical models and cell-free assays to validate each enzyme cascade. Example, a prototype shuttled starch converting enzymes from barley into coli, turning agricultural waste into bioethanol, our microbial distillery for renewable fuel. Controlling growth and division. Kill switch circuits halt runaway proliferation. We program cells to enter sleep mode under chemical inducers, or self-terminate if they escape contaminate. Case, MIT's 2017 kill switch triggered coli death in the presence of the arabinose, your safety net against microbial misbehavior. Applications in biosafety, from direct delivering nanocells to environmental bioremediation agents. Synthetic cells are poised for commerciation, yet without rigorous biosafety protocols. They are potential biohazards, always design dual safety locks, physical containment, and genetic kill switches. Artificial cell design is synthetic biology's summit. Constructing life from scratch demands both audacity and ethical caution. Let's push beyond carbon-based life. Could we craft silicon-based living machines? Silicon atoms, like carbon, form 4 bonds. What if we build polysiloxane chains that mimic cell membranes? First of it, fundamentals of silicon chemistry. Silicon oxygen, backbones abound as silica and quartz. Lab-synthesized organosilicon compounds give us elastomeric networks as membrane analogs. Imagine a silicon shell flexing like a living skin. Definition: polysiloxane polymers act like Teflon armor, resilient, biocompatible, and tunable. Silicon-based information storage. Beyond DNA, we explore peptoid oligomers and silicon-based polymers as data carriers. Stable at high temperatures, resistant to nucleases, we are designing bioinformatic systems that survive volcanic vents and cosmic radiation. Silicon-based catalysis and metabolism. Natural enzymes handle hydrocarbon chemistry. Silicon analogs need synthetic active sites. Tonight, peptide selenase catalysts to show promise, breaking down plastic waste into reusable siloxane monomers. Example, silicon-empowered catalysis have begun to degrade polyethylene, terephthalate. Imagine reversing centuries of plastic pollution with microbial factories. Bioeconomy and ecological impact. Silicon-based living machines could produce materials without tapping into ecosystems. A circular biorehineum cycle recycles waste into new resources. Yet an escaped silicon cell could disrupt natural silicon cycles. Dual kill switch designs, physical and genetic, are mandatory. Silicon life strands at the border of fantasy and imminent reality. Will we cross into a post-carbon biosphere? Only time our bioethical resolve will tell. Synthetic biology and artificial cells would disrupt every sector, from industry to healthcare, agriculture to extraterrestrial outposts. Industrial biodesign, custom microbes converting waste into biofuels, biodegradable plastics, and specifically chemicals, carbon neutral or carbon navigate processes rewrite manufacturing. Medicine and personalized therapies, smart drugs deliver cell homing-on tumors, synthetic microbiome modulators restore gut health, and on-demand bioreactors produce patient-specific antibodies. We are on the verge of curing chronic disease with living therapeutics. Agriculture and food tech, engineered soil consortia optimize nutrient uptake, reduce fertilizer runoff, and ward off pests, yield amplifiers without ecological collateral. Synthetic probiotics in plants becoming living biofertilizers. Space life support. This is so significant. On Moon or Mars bases, closed loop, bioreactors produce oxygen, recycle waste, and grow protein-rich algae. Artificial cell power plants will be our lifeline on multi-year voyages. Synthetic life is humanity's greatest opportunity and greatest responsibility. Our deep dive into the next great molecular breakthrough, synthetic life and artificial cells, draws to a close. I hope this journey through carbon-based marvels and silicon horizons left you both shocked and accelerated. Coupon code Brainwaves. Use this on Udemy for SAT Biology 101, Unlocking Life's Blueprint, Level 1, and download your 3D printable artificial cell model to kickstart your home lab. This channel is the first launch of my molecular biology documentary dream. Subscribe, share your thoughts, and join me next time for life's extreme environments, astrobiology, and extraterrestrial microbes. We'll unearth organisms defying Earth's limits. You won't want to miss it.